think with no further ado, we should just go into the program. It's really exciting to learn more about WAVE's study in Huntington's disease. It's been going on for a little while. So it's really good to have this update today. And it's a pleasure to have Dr. Ralf Reilmann present about the project. He is really an experienced man in the Huntington field and in doing clinical trials. So that's amazing. And also to have Danlin Xu and Marissa Volpe from WAVE here. There are several people from WAVE, and I'm sure they will all help us answer questions at the second part of this webinar. So please, Ralf, tell us a little bit more about yourself. That was a very brief introduction from my side.
Yeah. Well, thank you, Astrid, and thank you everyone for inviting me here, Astrid and Philippa for the EHA, and also, of course, WAVE. I'm honored to present this study. I have to say that personally, it has been an exciting journey for me to be involved in the WAVE program since many, many years. I think it is one of the most exciting programs we're currently doing in Huntington's disease, and I may be able to tell you a little bit about why I do think this today and share some of my thoughts with you. I really would like to take you along on trying to understand why we do studies like the WAVE study, which I think has some specific features. You've heard a lot already in recent weeks, those of you who have attended the other webinars, about the basic ideas of lowering huntingtin, but I want to show you some specific issues that I think are quite exciting about the program we're talking about here in general for the future and for trying to treat HD. I have, as Astrid said, myself been involved in Huntington's disease for many years. Actually, I grew gray hair working for Huntington's disease. I started as a 23-year-old, looking like most of you, but it wasn't actually causing me gray hair that we didn't make the progress that we wanted to. Actually, I think, being able to look back for 30 years, I think we made amazing progress in working on HD. It was pretty much the time when the gene was discovered that I started a PhD in Huntington's disease. And I'm so honestly, I'm honored and I'm pleased that really by chance at that time, actually, I ended up working in Huntington's disease. I think it's an amazing opportunity for trying to change the fate of the patients because we have the genetic background of the disease. And I think we've been learning over the last 3 decades that we made so many progresses that we can really now look into effective treatments. So I was pleased with my group to be able, you see it in the background, to actually open a private research institute, the George-Huntington Institute, about 10 years ago. So we do a lot of clinical trials in Huntington's disease. We do just about every trial that is running. And I have had the privilege to be involved in multiple international programs, including committees like the EHDN and the HSG Executive Committee and multiple research projects. But I don't want to talk too much about this. I may tell you a little bit as we go along. I think I should start sharing the screen. Let me just see. Can you see my screen now?
Yes, it looks really good.
Okay, good. Thank you. So it was worth changing the monitor.
Yeah.
So I already mentioned some of my disclosures. I think I should still mention that I've been involved with WAVE since many, many years. As I told you earlier, I'm also part of the Huntington Study Group, and WAVE actually started the program in North America, and I was privileged to being asked by them early to join their clinical advisory committee. I've looked that up actually yesterday, and it was 2016. I couldn't believe it. It's been 4, 5, 6, 7 years ago. And then when the study started in 2017, I was asked to join the dose escalating committee, which we will mention a little bit later maybe. That's a committee that reviews all the data once a certain dose has been applied to patients for safety, and then escalates the dose if things are safe. So I'm involved in the program for quite a while. It's been exciting for me. As I said earlier, I've been involved in the field for so long, but with the gene being discovered, I think the opportunity for us was arising to actually look at the roots of what causes Huntington and what causes Huntington's disease. And one of them obviously is the gene, the huntingtin that you know. And I think it's been enormous to see what progress we made over the years. And I think on the way to a neuroprotective disease modifying therapy, we've been doing multiple steps now. And it's my personal hope that the bridge will be finished soon in terms of construction for a therapy that will really change the disease. As you know, so far we can do something about the symptoms, but not really change the cause of the disease. Nevertheless, I think most of you know that one of the core hypotheses, one of the core ideas how to treat Huntington's disease these days is to work on the level of trying to reduce the production of the huntingtin, which is the protein that's produced by the Huntington gene, that you see in the lower side here. Actually, do you see my pointer if I move it? You can see it?
Yes,
right?
we do. Yes, we do.
The huntingtin gene is really like the construction plan of huntingtin, which is important. We all have it, also healthy people have it. And in order to produce the protein from the construction plan, you need a messenger RNA, a copy basically of the gene's instructions. And that is used to then produce the huntingtin in the cells. So the so-called, often called gene silencing approach to huntingtin would be to try to lower the production of huntingtin. And that can be achieved by using genetic molecules that are actually using the idiosyncrasies of DNA. DNA likes to be assembled in a helix. You have one chain and another chain, and they glue to each other if they're complementary, the parts that make it up. So you can build a little thing that actually is complementary to the huntingtin RNA, to the construction plan, and you can block it and it can be degraded. That has been shown in multiple animal models, and it actually does work. You can reduce the production of huntingtin this way by degrading the construction plan. But let's start a little earlier in the whole thing. Let's review, because I think this is extremely important for you to understand why the WAVE approach, in my view, is such an interesting approach to treating Huntington's disease. Let's review a little bit the genetics. I really want you to understand that. It's not a no-brainer. Well, you know, we always have two copies of any gene. That's important to remember. So the huntingtin gene, actually, every one of us has it twice. Everyone, also the healthy people, everyone has the huntingtin gene. So in this graph here on the left, you see a Huntington patient. In this case, it's the male in a couple, and the female partner here is unaffected. You see there is one gene that has the mutation, the CAG repeat included, and the other allele, however, is healthy. So the Huntington patient has two alleles. One is affected with the mutation, and the other one carries a healthy huntingtin. We need to keep that in mind because that's important today. It's very important for the WAVE therapy that we have a healthy copy, everyone. Also, those people of you guys listening now suffering from the disease have a healthy copy, and we can hopefully make use of that. That's important to understand. So the healthy person here in this couple doesn't have that, but what we see is that if we pass on, if we get children, very different combination opportunities that we have here. If the affected father gene, the father one, is combined with mother one or with mother two, we have the carriership of the Huntington mutation in the next generation. So these two children would get Huntington's disease eventually. If, however, the healthy gene is passed on and combined with one of the healthy genes of the mother, then we have healthy children. So that's the 50-50% inheritance chance. Remember, you have a healthy copy also in those people affected. That's important. Only one of the alleles that we have is mutated, and always you have a healthy copy. However, it is enough to have just that one copy to get sick because Huntington's disease is dominant. That's how this works in genetics. By the way, actually, I want to briefly mention this. I think this pattern here that shows the distribution of the genes from the parents to the children is something that's really, of course, very much a core feature of Huntington's disease. And it's included in our logo. One of our gene carriers from the registry studies when we started at George-Huntington Institute, he's a graphic designer, did that for us. It quite moved me a bit because I think it shows how much of a bad thing this genetic feature actually is. But then also I want to show you today how much of a chance it in the end has for developing therapies. We all remember that the CAG repeat is what is altered in a Huntington gene, right? So we have this, everybody has a CAG repeat in the Huntington gene, but in Huntington's disease, it's expanded. And the longer it is, the earlier the disease starts and the faster it progresses. So it's important to understand that. For treatment trials, we know that Huntington can actually be aggregating. And luckily these days, we've been able to actually develop methodologies to measure Huntington. Now, why isn't that running now? It did run before. So unfortunately, the video seems not to run. It's been working earlier when I tried it. Anyway, we can measure the Huntington concentration these days very precisely. And that wasn't the case 10, 20 years ago. So we've been learning a lot over the years. We've been able to much more precisely measure Huntington in cerebrospinal fluid or the blood. And that's important because we'll see that in a second. Well, let's see if this video works. It doesn't work either. Anyway, we see over the time that the Huntington brains do also shrink. You know that there is a function of the altered Huntington that's somehow toxic. Something is wrong. Something happens that Huntington does what the normal Huntington doesn't do in healthy people. And that's what causes Huntington's disease and all the symptoms in the end. So there is there's been a big progress over the years to characterizing Huntington's disease protein. This has been work done by Stefan Kochanek, a friend of mine from the University of Ulm. We know exactly now how the structure of Huntington looks like. And interestingly, if you show this a little easier in a way, you can imagine that the normal Huntington here, if we have a polyglutamine chain, which is induced by the CAG repeat, may alter its form. So all of a sudden, it looks very different, right? It's the same thing here that on the left hand and on the right hand, but on the right hand, we have this polyglutamine chain which squeezes in and opens up the structure. And now there are new areas of the Huntington gene that are all of a sudden available for metabolic exchange. And that's what's happening. You can compare this a bit to the sundew plant, it's called in English, I didn't know, but I was fascinated as a youngster about these things. You remember probably that they can eat little insects. So this is not dangerous at all. That would be the normally folded Huntington. But if the conformation is altered in this case now by the expansion of the CAG repeat, all of a sudden it opens up and there are new features, new functions that are available. And in this case, with this plant, it will be very dangerous because it can kill insects. So that's the gain of function toxicology hypothesis in Huntington's disease. And that's really the basic idea behind trying to lower Huntington as the rationale of the wave therapies as well. So now, however, caveat, Huntington is, as we have learned in the last years, extremely important for us. So the function of the protein Huntington is enormously important for normal life. Huntington interacts with hundreds of different proteins. We've been able to do that research. I'm not going to explain them to you, don't worry. But
it looks like a German roadmap, honestly.
Well, actually, I would claim German highways are less complicated to navigate than this one, even. But it's interesting. I think I just want to mention this to all of you, to the patients that don't usually do this kind of work, to appreciate how complex this is. Huntington has multiple functions in the brain and actually beyond. So we have also learned that if we knock out Huntington in mice, they can't deliver fetuses, right? They die in fetal stage, the embryos die. So Huntington is important for neural development. And if we knock out Huntington, if we kick it out genetically in mice in later stage in life, that results in severe deficits. So Huntington has some important role and we need to appreciate that. That's core importance to understand why the wave approach, in my personal opinion, is a very interesting and cool one. Because that's what wave is really doing. That's the essential message here. We usually, with most of the therapies that we've been testing so far with Huntington lowering, these approaches lower both the mutated, which is the red one here, and the blue one of the Huntington, so both. You remember we've been looking earlier at the alleles of the parents and the children. So you remember there's always a healthy one in anyone, including the patients. So wouldn't it be cool if, considering what we just learned, that Huntington actually has pretty important functions, it seems, in the body, if we could preserve the healthy huntingtin, right? It's there. Why lowering it if we can reserve it? Because there may be side effects otherwise. And on long-term treatments, particularly, it could have things that we don't even think about yet. So I think genuinely, naturally, when I heard about the WAVE program 8-9 years ago, I was fascinated by the idea I was fascinated by lowering huntingtin anyway, but going allele specific seemed to be a clear thing for me to be attractive in terms of trying to avoid side effects and being able to possibly, treat earlier. Don't have to worry so much about the dose because we can knock it down as much as we want. So in order to understand how WAVE approaches this therapy, I want to mention one biological feature. I can't go around that. There are mutations in the body, in the genes, which we call single nucleotide polymorphisms, SNPs. So what happens is that one letter in the DNA changes to another one. And because an amino acid, that is the basic backbone of proteins, can be coded by several combinations of 3 DNA structures here, actually, it wouldn't even matter possibly. It could be that the protein that is produced looks the same, but there is a change here at some people in the DNA. And that happens in multiple places. Happens so that some specific changes are located close to the huntingtin gene, and they are preserved in the populations. If we look at multiple generations, these single nucleotide polymorphisms, these specific changes in the lettering of the text, so to speak, are inherited. So they're there in populations. There are certain SNPs that are more or less frequent in Huntington's disease. And since the antisense oligonucleotides that we're using in this treatment can be very, very specific to certain areas, we can target these SNPs. So if you have a certain SNP on the allele that's diseased, that is the mutated allele versus the healthy allele, we can use that as a target for ASO therapy. And that's what WAVE is doing. So if you have a SNP that is on the mutated RNA, on the mutant allele copy, and it's not on the wild type copy, we can target that for therapy. We can have a little agent that clicks to this area of the RNA and degrades it. So important to know for the WAVE programs, for all of the programs that WAVE does, we have to do a little genetic test first because we have to find out what SNPs you have. And if the SNP happens to be on the mutated huntingtin and the wild type, we can actually not do the therapy because it would lower both of them. We can only do the therapy, we can only recruit you in a clinical trial if you have the specific SNP we're looking for on the mutated RNA, but there is a good chance it's there. And there are different SNPs that we can use for future therapies. So the approach is to use that SNP to anchor the ASO against it and then get the construction plan degraded. That's the basic core thing you please need to understand if you want to do this trial, because that's how it works. And again, important possibly to preserve the normal here, which would work this way. We've looked in a study in the United States with colleagues how often these SNPs occur. SNP1, SNP2 covered about 60% of the population. There are more SNPs that we can use in the future once the therapy has been established to be efficacious. The first 2 therapies we looked at were actually in SNP1 and SNP2. So there were 2 SNPs that were shown in cell cultures to lower huntingtin. That's the SNP1 agent. It lowers the blue line here, the huntingtin concentration of the mutated blue huntingtin, whereas the wild type is pretty much preserved. That was also true for SNP2. With these data, we went into the SNP1 and 2 trials that have been conducted in the past. And these 2 trials were pretty similar from the idea to what we are now doing in the trials. We first used single doses, we escalated them. That's always done in early development programs. And then we did a multiple dose, even an open label. I don't want to get in these details yet, but I want you to know that these trials happened. Why do I want you to know it? Because we had some preliminary data on these trials that are interesting, I think. So what we saw on these trials was, this is the example of SNP2 trial. It was the one started first, just because the ASO was ready earlier. We did see that the MHTT, the mutated huntingtin, was actually lowered a little bit from visit to visit. These were multiple doses applied though. but it wasn't all that much. You see, this is the 30% line. So it was like 10, 15%. It was going a bit up and down. It wasn't great, but we saw something there, which wasn't significant. But if you look at multiple visits, quite a few people, there was clearly something going on, versus on the wild type area, we saw for the first time in a study that the wild type didn't change. It clearly didn't change. So there seemed to be at least an allele-specific effect here. But actually, interestingly, the two SNPs, the ASOs that were used for SNP1 and SNP2, were pretty old ASOs. And the efficacy wasn't enough for us in the development program to say, hey, we want to continue with this. So we stopped these two programs. And there was another reason why they were stopped. And that's something I want you to appreciate too. It has to do with innovation and progress. So look at this here. I think being from Germany, please excuse me if I make reference to Mercedes-Benz, but this was the first car built by Mercedes-Benz back in 1885. I think we would all be amused using it today, but we would probably prefer the 1950 version. Some of us the 2023, right? So that's an electronic version. What I want to show you is that things evolve. There is innovation and progress. It took them about 100, whatever it is here, 140 years or so to be where we are with cars now. ASOs, however, are only around for about 20 years in research. And in clinical development, it was even less time. Nevertheless, in the last 10, 12 years, you can probably imagine, with all the studies going on, not only in Huntington's disease, ASOs are looked at and tested in multiple diseases beyond neurology, actually, a lot of progress has been made. And we have novel ASOs with much better features than they had them 10 years ago. So an ASO produced 10, 20 years ago, as the one used for SNP1, SNP2 wave, or for tominersen, the Roche Ionis ASOs, they are, meanwhile, I would nearly say, sorry, apologies, guys, but I would call them old timers because the biological development is pretty fast. So let's appreciate that there are evolving things in biology. And one of the ones that evolved was the way how wave is actually constructing SNPs, sorry, not SNPs, but constructing ASOs. So the treatment approaches they use. So they have developed a new chemistry for these antisense oligonucleotides. And this new chemistry has proven in animal models to be more and better brain penetrant. There are longer effects that would allow us to use lower dosages of the ASO, which seems to be good overall because ASOs may have some side effects. We've learned that, for instance, in the tominersen program, maybe something specific to it, but we don't know yet. There could be ASO-specific class effects. So it's good to be low in the dose. And it seems to be well tolerated so far. Also a big advantage in the SNP3 program now is that Wave actually has an animal model that has a SNP3. The previous developments were done without a specific animal model available to test the hypothesis of allele-specific lowering. Now here I will show you one graph because I think it's important for you to understand with the animal data that we generated or Wave generated in the SNP3 model. So we see allele-specific lowering, and left is the cortex, right is the striatum, both very important areas for Huntington's disease. And you see on the right-hand panel here where we compare the blue is the wild type and the red is the Huntington versus the baseline, which is 100% up here. You see that there is clearly allele-specific lowering at all these weeks over 3 months in this animal model here, and that's true in the cortex and in the striatum. So we see the new ASO, the SNP3 ASO works on that SNP. Again, the animal model had that specific SNP introduced. And we saw in the animal model is safe. It's well tolerated. It's actually been looked at in multiple animal models for safety, large animal models, and so on and so forth. So I think it's very exciting times for us now to be able to say that we now do the clinical trial since quite a while, the Select HD trial with the new generation ASO targeting the SNP3 specifically. And what do we do in such trials? It's the usual suspects. We look primarily in the earlier developments at safety and tolerability. That's always important. We use the UHDRS that the patients of you know from Enroll and other studies, a clinical assessment scale to look for also for safety and also some efficacy. We look for key biomarkers, the mutated Huntington, important. And by the way, ways to complement, we have developed a much better assay to measure wild type Huntington. That's not trivial because previously that wasn't possible precisely. It's very good to have that at hand right now to do mutated and wild type precise analyses to see what's happening. We are monitoring NFL. Some of you may have heard NFL. It's actually a marker for neuronal damage. So it's really good to have that monitored because it gives us a chance to see whether some damages occur somewhere and interact. And we look at the MRI. We've learned from previous ASO trials that there were changes in MRI volume, which we clearly don't want to see. We don't want ventricular enlargements for whatever reason. So the trial, the SELECT-HD trial, is looking like that. We started with 30 milligram a single dose. Then we looked at the safety. It was safe. We went to 60. It was safe. We went to 90. We're in that phase right now. And we hope to be able to proceed to multiple dose soon, then, which was done in all the other development programs in the SNP1, SNP2, and also with the Ionis product. It's a normal thing to do to first look at single dose safety and then multiple dose. Now, the study is actually ongoing in multiple countries in the world, also in Europe. I think this is our primary target here today, our target audience. So there are multiple countries in Europe, in Poland, Denmark, Germany, UK, France, Italy, and Spain. There are opportunities to support this trial if you want. But I just want to reiterate again, I mean, we're very grateful as the investigator community if you support this trial, but it needs to be tested whether we can actually include you by this genetic profiling. If this study is successful, however, then I would of course hope that for the future, there will be new studies for SNP1, SNP2, and whatever other SNP we have. In that context, I may mention that actually there are even studies these days of one single patient to receive one ASO that has a single mutation. So these studies, these experimental studies exist. They are being done by the German Network of Neurodegenerative Diseases, and there are multiple opportunities for the future. That's what I say. That's not Wave's statement, but I'm just saying there is a feasibility to be very optimistic for the future to actually reach out to multiple people that have SNPs, even if it's not a SNP 1, 2, or 3. So I'm grateful for Wave, to Wave for sharing this preliminary data, which I think is pretty interesting as well. So this is preliminary data from the ongoing SELECT-HD study. Was a lower number of subjects. You see on the left-hand side, placebo group, MHTT concentration in the CSF again, and on the right side, the treated people with 30 and 60 milligram. We didn't have 90 milligram data sufficiently at that time yet. But you see here that after a single dose, there is something going on here. It's going down. So that's the hope, of course, that even if we increase the dose because it's safe so far, we get even higher effects. Here we are at least scratching the 40–50% mark with some of these people. So since it's safe so far, we'll soon have the 90 milligram data, and it's really a well-tolerated trial so far, this agent. And the data does suggest that we are hitting an allele-specific mechanism. So getting to the end, so the real interesting issue is, I think, that it seems safe so far. It is safe so far up to 90 milligram. We've treated 18 patients with it. There were no serious adverse events at all in the study. No participant discontinued the study so far, which is good to see. There were some increases, one has to admit, in NFL. This is the marker for neuronal integrity. But we've seen that in multiple studies, and we have to look at the dynamics. This is carefully monitored by the Data Safety Monitoring Board, but it's all within ranges to continue the study, and we'll monitor this as we go along to multiple dose, hopefully. There were no signs of inflammation in the CNS so far, which is great and important because that's an indication that we don't have to think about certain side effects that otherwise could occur and possibly have occurred in other programs. So to summarize, I think it's really interesting to see that the early data also on the SNP3 program here indicates that the ASO is hitting the target. We might be able to go for an allele-specific lowering, which again, remember, would preserve all the normal functions of the Huntington. It's the one-sided thing and not the bilateral. That's what we want. I think we want to preserve function. There is activation of even new clinical trial sites because we wanted to expand the cohorts a little bit to get even more robust data from the measurements, and additional clinical data from this program can be expected soon, possibly at the end of the first half of this year, and that will inform future decisions and the further development of the program, hopefully to phase 2/3 trial, we'll see. So we don't know where this road is taking us, as always, but I think to get back to my initial comments, I believe it's amazing to see where we are today and that we have the opportunity to tackle the questions. Can we do something to improve Huntington's disease by going to the roots, by changing the protein concentration? Certainly, I believe that an allele-specific approach has a lot of possible pluses for introducing a therapy like this early and for a long time. And I hope we are successful in one of our programs, and certainly the WAVE program is one of those that I would like to see succeed and give an option for therapies for Huntington patients in the future. So thank you for your attention, and I'm happy to answer any questions. And I'll stop sharing my screen, I guess.
Yeah, maybe that's good, so we can see each other.
Yeah.
Thank you, Ralf, for this presentation with so much information and really interesting. But I wonder, could you just very briefly say when did this study start to recruit? How many, and for how long are they treated in the trial?
Yeah, I mean, the study.
Maybe I missed it. Maybe you said it. Maybe I missed it. I'm sorry, but.
No, the study started some time ago, and it is actually an issue, of course, to see to select the people with the SNP3, because not everyone has a SNP3. So that's what I mentioned earlier. You have to be prepared But
do you know how many are we looking for? How many is going to be included in the trial? Is there a fixed number for that?
Then can you answer the final question, because we've changed the numbers quite a few times?
Okay. Maybe WAVE can answer? Yeah,
WAVE can answer the final numbers right now.
We have enrolled 18 patients for three dose cohorts. Each cohort was 6 patients to start with, and the randomization 2-to-1. So 4 patients received active drug and 2 patients received placebo for each dose. And our study design is adaptive. So DEC is review blinded data, and SMC reviewed unblind data. And based on the SMC recommendation, we're currently expanding our dose cohort, the 3 dose cohorts, to more patients. So we are expanding our single dose cohort to get more data to provide us more informed decision moving forward.
And to answer your other question, Astrid, there is the duration of the study, which is always an issue, of course, for participants. We have the single dose, the initial dose that we apply. And then, as I showed in this graph, we are waiting until we have the safety from multiple cohorts to go to multiple dose. That's probably going to happen soon. But then you would receive 3 additional injections in the program. So it would probably then take another like 4 months, 5 months. But the overall duration is a bit individual for the subject. Those that start early have to wait a little bit until the multiple dose comes. And then it's kind of restarted with the multiple dose phase. We do that for safety reasons. We want to really see that we only go to multiple dose if we see good involvement of target. And as I indicated earlier, it seems to be the case now. So I do expect that if there are no tolerability issues, which, as I said, there weren't any yet, we didn't have any SAE or discontinuation, that will probably happen soon.
But if I understand correct, because I'm sorry, I'm a simple person, 18 people have been into the study and given either placebo or 2 different doses one time. And now we are waiting for the results to continue with 2, what did you say, 3 or 4 injections? So because this is spinal, what's it called?
Lumbar puncture, yeah.
Lumbar puncture,
the same that we used in the tominersen generation HD1 study, or in the SNP1, SNP2 studies of WAVE, or in HD Clarity. So we need to give the drug through lumbar application, because it needs to penetrate through the brains this way. The chemical compound cannot be injected in the blood or in any other way. No, we want to expand the cohort right now to get more data on single dose, and then it's going to be a multi-dose phase for everyone that is now included. So for me, always, I have to say, I see this as one big program, like the SNP1, SNP2, SNP3 studies, and these 2 studies already had quite a few subjects included. And now it's the SNP3 study that needs for this specific SNP, because of the new form of the ASO, the support to see whether this new generation ASO is really well tolerated and works more efficiently as we hope. Yeah.
Okay, so a little bit more complicated in a way than we are used to, I think, but very, very interesting. So Philippa, sorry, I will give over to you to see if there are anything to pick up on from.
Yes. So we have 3 interesting questions in the chat box. So the first one is a question by Dina, who asks about what is the percentage of the population covered by SNP3?
So based on the publication, the SNP3 is about 40% among all the Huntington disease patient population. We are actively screening patients to see whether our number matching the publication. So based on publication, around 40%.
Did you say, sorry, just to pick on that, because that's about the inclusion. So you need to have this SNP, which nobody would know. You need to, of course, do a genetic test to do that analysis. But what else are the criteria? Is it early stage disease? I'm sorry, maybe I missed it again, but.
Yes, early stage. Yeah.
Yeah, it's an early stage disease. And I think, it's always a little difficult to give these data here. I think you should refer to your center and ask your neurologists about these specific criteria. But what we really want to see is the people with low disease burden, because that's the phase of the study, unfortunately, for those people that have a bit more progressed disease at this stage. But that's the phase where we can establish the efficacy of these therapies much better. And then if they are efficacious and eventually approved, they will hopefully be given to everyone. But nevertheless, for the research purposes, it's important to look at certain criteria here. They are specified and known to the centers, but it's more the early spectrum of people that have some symptoms, some slight symptoms, yeah.
And are these SNPs determined by a blood test?
Yeah, we draw blood, and actually this has been also improved. I mean, this is of course a new area for everyone involved. We've had big improvements in the timing of these assays in the earlier studies, SNP1, SNP2, that sometimes took quite a while to get the results back, but now it's getting within 2, 3, 4 weeks, we have the results. And the way this works usually is that if somebody is interested to be in the trial, of course, the centers also reach out to subjects that they know from Enroll HD or so that would work. Then we look for the inclusion criteria after the patient has been, is interested, and then there is an informed consent signed. We check everything, and then the blood test is done at a very early stage at screening. And we look at whether the blood fits, whether the SNP fits or not. And if it does fit, then you can really be included in the trial. So as I said earlier, I would like to encourage you to support a program like this, which will give you a chance of being, unfortunately, not able to join this particular program, but you would support even the programs for the future. Because if the SNP3 is successful, I would be very surprised if there wouldn't be new programs for SNP1, 2, and other SNPs in the future that would eventually cover pretty much the range of Huntington.
Thank you. So another question is about how should people that are on non-allele specific trials interpret this data that you presented?
I guess I should take that. I don't think that this is of concern right now, because we haven't shown that the allele-specific approach is clinically better or that it is even efficacious. All I'm saying is I think it makes a lot of sense to explore whether allele-specific lowering is possible and feasible. And if it is, it seems to me quite compelling to say, why would I want to risk to lower a protein that has so many important functions? Those people enrolled in other programs should continue in those programs for now, and we will learn what this means. But we have to be aware of the fact that everything we are doing here, the lowering of the Huntington protein, is an experimental approach to trying to treat Huntington's disease. And at this point in time, we don't even know if it actually tackles the basic mechanism underlying the changes. We do think it is a good opportunity of treating Huntington's disease, But I'm sure many of you in the community have also learned about things like somatic expansion and other issues that are ongoing in Huntington's disease. We have to see which one is the right approach to treatment. Maybe it's a combined approach in the future, but I do definitely 100%, 200,000% support that, you know, testing the hypothesis of protein lowering properly now is exactly what we can do now and what we should do now to advance the science to the right direction. Because if it does work, as I said earlier, then I think it will be possible to find approaches to lowering specifically, and there may be, you know, future steps possible to make that even less burdensome if we know this all works. There are multiple things to discuss here, but we can't do this here.
Another interesting question was made by José Luis Sendon from Madrid. He asks, is there a chance that normal huntingtin lowering had a role in the toxicity of the other trials?
I mean, that's what I kind of alluded to a little bit when I briefly mentioned this earlier. I mean, I'm aware of the fact that this is talking about the WAVE study, but being an investigator, I think I have the liberty to kind of put this into my framework of thinking. And if I do this, I have to say that at least, you know, we haven't understood yet, I think, at this point, to the degree that we would like to, what happened in the tominersen program in the Generation HD1 study, for instance. But it is of concern that we have a ventricular enlargement that we saw in these subjects, and it was dose dependent, and there were clinical effects that weren't looking good. So we have to learn, we will learn, because there's a new trial coming, we will learn whether specific populations, the early stage people, don't have these signs. We'll learn this, but at this point in time, at least for the somewhat more progressed people, it seems no alternative and no option to use tominersen, for instance. And yes, it could be that there is an on-target side effect that we saw. I think a bit more about that since I learned about the Novartis data, which has shown us also with the branaplam oral protein lowering approach, that there has been also ventricular enlargement in these people. And I haven't seen all the data yet, but it's speculation at this point in time. There are multiple possibilities that could have caused that effect in the branaplam study, but one of the possibilities to consider indeed would be to say, well, it could have been an effect of on-target, non-allele specific lowering of huntingtin loss. Why? There is a reason for this for me to think about that, and I didn't have the time to show this today. There is good evidence for huntingtin to have multiple functions. I showed you this big network thing, but there is one function that is pretty well established, and that's, for instance, the effect that huntingtin has on cilia. And cilia are little hairs that move around on different surfaces of the body, including the internal skins that cover the area where the CSF is floating. So there are cilia that actually are in part responsible for the flow of the CSF. If indeed, it's a hypothesis, but if there would be an on-target effect of lowering huntingtin to a degree where maybe the ciliary function is impaired, that could explain why CSF hasn't been transported properly and was blocked to get absorbed again, where it physiologically should be absorbed in the upper areas of the skull, and could theoretically be responsible for the ventricular enlargement that we saw. But it could also be immunological reactions to the tominersen in the case of the ASO, and maybe something totally different, an off-target effect of branaplam. We don't know that 100%, but it is true that hypothetically, at least, we could think that it is a non-allele specific issue. And that, again, we can learn from WAVE. If the WAVE program would show there is no ventricular enlargement, it would be another indication to believe, well, maybe something is going on there. That's how science works in the end, but a very good thought.
We also have a question from Freddie Hall about the concerns. Are there concerns with the observed NFL increase, given that several trials were discontinued, as you said, because of issues like this?
Yes, I mean, personally, the WAVE people can answer as well. I personally think, yes, we have to be alert to this, but again, so far, it was nothing that created any clinical program consequences. If we see that there are clinical consequences or side effects like ventricular enlargements, things will change differently. but we haven't seen anything like this yet. And it's definitely, I think, worth exploring this, because the level that we saw was, again, still tolerable, because otherwise we wouldn't have given the permission to expand the dose and expand treatment. And we can see NFL increases because of multiple things. But indeed, this needs to be monitored. However, a little caveat also from my end, I have seen a lot of clinical trial data in the past few years. And I would like to encourage everyone, if you see data of mutated Huntington protein measurements and NFL measurements, look at the variability over time. Don't look just at the end of the day, so to speak, at the baseline versus end of trial. Look at the variability of the assessments. And I think we still have to work on the quality of these assessments as well, these markers, these wet biomarkers. It's not trivial. So not everything in terms of numbers expressed there might be one-to-one translatable to clinical effects. I'm very grateful for the community to have developed what we have in hands right now, but we shouldn't take everything for 100% granted and 100% conclusive there. We have to look at the data carefully, how the data has been collected, how it's been transported. These are very sensitive issues. And I think that in the very end, the core thing that we need to look at is the clinics. And if, as in the tominersen programs, we see TFC is declining and things like that, that's what really pulls the thing. But if there is some imaging change or some biomarker change, we have to carefully be, polite and say, well, we don't want to miss a treatment option here because there is some effect on a biomarker that we haven't really understood yet. That's the reason why regulatory agencies don't accept things like MHTT or imaging or NFL as an endpoint for a clinical trial. They want to see clinical benefits.
Another very practical question, this time from Vlado from Slovakia. He asks, if this works, how long will we have to wait for this therapy to be available to everyone?
Well, if I may take a big quick shot on that before the WAVE people may be answering as well, it will take time. You all know that. I mean, unfortunately, that's nothing that will be available tomorrow or the day after tomorrow. If this program is successful, though, and in that sense, I would think at this particular time, it would be, I think, because there are some studies ongoing, as you all know, that we all want to support, the PTC study, the whatever studies is ongoing, I think they're all important. But I think this particular allele-specific approach would be nice to know soon, because the sooner we know whether this approach works on the SNP3, the earlier we could expand again to other SNPs and get to a phase 3 study. A phase 3 study will take a few years, so be realistic. If this really has a chance, talking a few years, but we've seen how fast it went from doing the Ionis phase 1 to getting Generation HD1 results and getting the new study started. In the end of the day, I think we are on a good, very, very good path. And we are only on that path because you all help us. So in the end of the day, please support all these studies, whatever you like personally the best, and then we get sooner to the point. And I still want to live to the point where I see a treatment for Huntington's disease, right? So let's get it done.
Going back to the SNP again, Dina asks if it would be a good idea to have your SNP profile done in your Enroll HD visit. What are your comments?
Actually, I've been thinking about this for a long period of time, and I was nearly going to say earlier that in the future, maybe indeed, if we do genetic testing for Huntington's disease, we should get all that data in. There are always things about confidentiality and all these things, and I'm not an expert on that. But I would think in the future, I could well imagine that we will be looking very carefully at all the targets that have been identified as possible genetic treatment targets, if that's the case. And then, of course, we would be able to tell you right away what options would be available to you. It's also, don't be mistaken, you could have SNP1, SNP2, SNP3, you could have multiple SNPs, and then you can even choose whether you want a therapy for SNP1, SNP2, or SNP3. The important thing is whatever SNP we target, it needs to be associated to the mutated allele, and it can't be on the non-mutated allele. Well, it could be, but if it would, then we would do non-allele specific lowering because we would lower both of them. We don't want that in this program, at least not in the experimental program. But Dina, I think this could be solved, and actually quite easily solved, because that could be done in the future.
Good. Shelley, you want to have a final remark?
Just thank you, Astrid, to you and your team, and Ralf, to you for sharing your perspective on our study. I think you said it well, as in terms of we can't do it without all of you. So really appreciate your support, and Astrid, appreciate your call to action. I think you delivered that very clearly. So if there are any questions, certainly feel free to reach out. We're here to help educate and really bring awareness. And hopefully, like you said, the faster we move, the faster we'll know more about whether or not this approach works. And so, yeah, thank you all very much for your time today.
Thank you, everybody. Thank you.
Yeah, thank you. And really, I think what Naeem is picking up in the chat, also representing Turkey, consider for the phase 3 where to go and how can we work together to have this recruitment potentially for a phase 3 with larger numbers of patients, and work together in having that done as fast and efficiently as possible. But thank you so much, Ralf. Thank you so much for your excellent presentation and super interesting discussion as well.
Thank you for the opportunity. It was a pleasure for me to be able to share these thoughts with you. And if anybody has a question, don't hesitate to reach out to me. Thank you. Good luck.
Thank you all.